Hello everybody and welcome to BioStock Live from Stockholm, Sweden. Today we have an interesting program. We have 3 companies here to present to us today, and we are going to learn all about digitalized pathology, and we are also going to hear about prevention of thrombosis. And not the least, we are going to hear how you can make bone graft transplant substitutes from reindeer. So quite an interesting program, and I'm very pleased to welcome today's first speaker, who is Stefan Nilsson, CEO of Lumito. Welcome, Stefan.
Thank you. Hi, I'm happy to be able to present Lumito for you all. My name is Stefan Nilsson, and I'm the CEO of the company since 2 years ago, when the company started a new strategy for building products out of its IPR. So, what are we doing? We are actually changing the way you can do tissue diagnostics. Today, you can see the image on the right side of the slide here. That is the normal way of how tissue is presented, tissue information is presented to pathologists. It's an H&E staining, hematoxylin eosin staining. And as you can see, the information, there's a lot of background information, and when you're looking for specific tissue, malign tissue, it can be difficult to find that with all those information. And that's the reason for when they made the studies of this, that it's you can have inter-observer agreement for of 70%, 70-75% when doing diagnosis of tissue samples. And of course, it's not the very obvious ones where there's a lot of expression of for instance, cancer, or the very where there's very benign samples. But in the middle, it's even less than this 70-75% when it's atypical tissue or pre stages of cancer, for instance. So that's what we are aiming for, to improve that and to make high-end the accuracy. A brief history of the company. The company was founded by researchers in Lund University in the early 2000, and by researchers led by Professor Stefan Engels, and that's the Institution of Atomic Physics in Lund. Based on the research, they started the company in 2010. And as I mentioned, the strategy was changed in 2017, where at the same time I entered, and our CTO, Anders Sjögren, with the goal to do products, develop products out of the IPR. The patents here is I will come back to that, but that is basically imaging based on a specific form of nanoparticles called UCNPs, up-converting nanoparticles. We started in 2017 by doing a new product plan, strategy, and funded that. And the proof of concept started in early 2018. And now we are in the middle of the development phase of the first products, which we are aiming for launching in the first quarter of next year. So as you can see, we are moving quite fast within this industry with diagnostics instruments. This is quite fast. So our focus, we have focused on tissue diagnostics and medical medtech applications, where tissue diagnostics is the first form. As I said, we were basing all this development on the company's IPR. We have patents in the main markets approved, and also patent applications for specific applications such as immunohistochemistry. And of course, we are a part and wants to drive the track towards digital pathology. Today, digitalization within pathology is not very high. And there are certain challenges, which I will come back to, that we also address. How could we do this in such a fast manner, since we started with mainly the patents and the research in 2017? We have made a setup where we outsource all development to our partner, TTP, the Technology Partnership in Cambridge, UK. So they are doing all the development work on our requests. And that made it possible for us to start directly as soon as we got funded and do the proof of concept phase, and also to scale in a way that is not possible otherwise. If we were supposed to build our own development department, we would not be out of the proof of concept phase today. So that has been a real success factor for us. So we are keeping the core competencies in the company, but we are doing all the development work with our partner. So what is the reason for doing the tissue diagnostics? When we looked at this, we saw that the market is increasing with 10% per year. And the reason for that is that the aging of the population, basically. The demography is people are the average living years are increasing, and thereby also the sickness level. So that's one. The second one is that treatments are now more specialized and individualized. So to be able to do very precise treatments, you need very precise information about the indications, and the diagnosis must be very precise, and therefore more samples and diagnostics are made. So and the third one is that in the third world, developing countries, the living standard is increasing, and therefore people can afford to be diagnosed and have treatments. One of the very important reasons for this is also that pathologist is a limited resource. So the ability to diagnose all these tissue samples is not increasing along with the demand. So there are lots of drivers that for a method and the workflow that could increase the throughput in the system. So our goal, we are doing this in two steps basically. The first step is to provide a system and kits to improve accuracy, so that the pathologists themselves can have better information and do more accurate diagnosis, and also be able to multiplex within the same samples, the same tissue sample for different indications, and also multiplex with the hematoxylinosin stainings, as you see in the first image, and by that we can reach the market very fast and can get our technology out to the labs and to the pathologists. The second step is, with our technology we are able to do quantification, since we can measure the light from the nanoparticles, and thereby do quantification of the expression of, for instance, the cancer cells that is in the sample. That is very difficult or impossible to do with the current staining methods available today. And that is also important for the pathologist to when they're doing diagnosis. So what is it we are doing that is specific? We have these nanoparticles, which is the big let's see here, there we are. And this is a very general description of this. We put, for instance, biotinylated antibodies on it. Those antibodies stick to the antigen we are looking for in the tissue sample. And therefore we have nanoparticles stuck to exactly what we are looking for. We stain all the sample with this, and then we excite with laser light on the sample. And this specificity of these nanoparticles are that if you excite them with a certain light, they will send out light with a shorter wavelength. Everything else in the sample, when illuminated, will be send out light with longer wavelengths, and therefore we can really see the difference between our particles and the rest in the. So all the background is basically removed. So you can have the analogy with the needle in the haystack, that we remove the haystack and you see only the needle in the sample. So this is the kind of images we get. So here you can see this is tissue from colon that we have stained with pan-cytokeratin, and you can see that you don't see the background, you just see the structures of the cells that we are looking for. So this is so if there were no such cells that we were looking for, or then these images would be completely black. So this is just to compare, and this is very obvious. We have done hematoxylin staining together with our particles, and you can see that the background is removed, but this is also to show that we have specific staining around the membrane here of the epithelial cells. So this was basically what was done in the proof of concept to show that we have specific staining with our method. These are cultured cell lines for breast cancer, and where we have stained with our particles. And as you can see, we have very high resolution, and you can see very well the structures of the cells. And it's also co-stained with so-called DAPI, the blue things, who stains the nucleus of those cells. And these are for another indication of breast cancer, HER2, which is Herceptin marker, and also you can see the same thing here. So it's very specific and very clear what you have. I was told a little bit about the tissue diagnostics market, and it's growing by 10%, as I said. The two big markets are North America and Europe. Europe about 30%, and North America about 40% of the total market. And we are going with a strategy for Europe first, since we will be able to launch our products very quickly on that market, and then North America as the second market. Roadmap, I have been into that a little bit. The roadmap, we are in the middle of the development phase, and we are optimizing both the instruments and the reagent kit. Our product is of course the instrument where you can view and make images out of the tissue samples, and also staining kit where you stain the tissue sample with our particles. So that is so we will have both an instrument and recurrent sales of reagent kits. That's the business model. And we will go over to the third phase in this autumn, and to be able to CE mark and set up production and transfer to production and stuff. And we have the aim of launching the first products in first quarter of next year, the Q1. So this is just some pictures of this is the alpha prototype we have built, and this is an indication on how it can look like in Q1 next year. So we are doing the alpha prototype ready now, and the beta will be in end of Q3, and then we are going for approvals and production. This is the only Swedish slide here. We are in the midst of signing period for units that is that will cover to develop the products and over the product launch. So with the end of signing period in the May 31st. Yeah, I think that was about it. Thank you.
Thank you very much, Stefan. So this sounds like a sort of a revolutionary technology.
Yes, there are lots of where you try to use those H&E stainings and, for instance, do artificial intelligence applications or machine learning approaches to increase the accuracy of the diagnosis. We are doing the opposite. We are trying to highlight what we are looking for and reduce the background, and then to do automatic analysis in the step 2, as I indicated earlier. So it is our goal is to really revolutionize pathology.
And in which area specifically, isn't as far as indications go, or different things, where do you see the biggest potential?
It's to diagnose cancers.
Yeah.
That's a different kind of cancers. So we are using markers that we are not developing the markers, the antibodies. We are using commercially available antibodies that is already used in the pathology labs, but we are like sticking the UCMP, the nanoparticle to them, and by that we can reduce all the background and make it possible to improve accuracy.
And going forward, presuming that everything goes well with the development and so on, you have a market launch plan, of course. When introducing a new technology, like especially if it's revolutionary, there are always challenges. What do you see as the main challenges for Lumito in that aspect?
Of course, the conservatism. Of course, it could also be that when coming with a new technology, you have to prove yourself, and that will take some time. So our approach will be to find reference laboratories where we can work together with as the first customers, and to get quick results out of that we can use for information to inform the society, pathology society, about our products.
And are you, as of now, in discussions with any specific clinic?
Yes, we are in discussions with several parties for that. Yeah. So everyone, of course, want to see it. So we now when we have the alpha prototype, we can do some presentations and demonstrations as well.
Okay. What about the competition? I mean, of course, the standard technology used today is the main competitor, you could say.
Yes, of course.
But are there any competitors within the same field?
No, and we haven't found any that has the same approach as we do. Of course, there's a lot of research around those particles, for instance, but our IPR is mainly covering this. As I said, artificial intelligence applications. That's the main competitor, I would say.
Okay. We'll see if are there any questions from the audience today. Yes.
Would this procedure be significantly more expensive than the present day technology?
We are optimizing at this moment, and our goal is to come down so that it will not be significant. And indications we have that is possible, so that we can come in within a range that we will be slightly more expensive. But in the other hand, we can do co-staining in the same tissue samples, which will reduce the number of samples. So you could do a good calculation on that.
Of course, cost efficiency is an increasingly subject that's being discussed a lot within the healthcare right now. Yes. And who would you say are key to convince here in order for you to get a really good market uptake?
That's it's the head of the pathology department, because if you can't go through there, the procurement will not buy from the hospitals, for instance.
Yeah.
So that is the key person or key persons.
Yeah. We have another question.
What is a future business plan? How to sell this? Are you going to sell the product? Are you going to sell the license for the software? Are you going to sell based on the numbers of they're doing or whatever?
As a first, we will sell ourselves first for the reference customers and the first early adopters of this technology, and then we'll go for a distributor strategy. And we will. We have adopted our system, so you don't change the workflow within the labs, for instance. So the labs are used to buy instruments and buy reagent kits, and we will adjust to that at least as a start.
Well, yet another question.
More of a comment. Where I see the future is the automatization,
yes.
Because everyone knows there is a severe lack of pathologists. So you must get quicker through flow of material.
Yes, and we see that too. So that's as I showed you, the step 2. It's quantification and automation. And based on our technology, the images are with higher contrast and no background will be easier to do a automated system for that than the ones you saw, the hematoxylin with a lot of background. So of course we are aiming for automation. But as a first step, we let the pathologist have this as a additional tool for when they're doing their diagnosis. That makes it easy for us to certify the product and get it out on the market. And in the meantime, we will develop the automation.
We are actually running out of time, so you will be able to pose your other questions to Stefan later.
Yes.
But for now, thank you very much, Stefan.
Thank you.
And with that, we are preparing to go forward in the program. Our next speaker is Paul Watkins, who's from BBS Bioactive Bone Substitutes, which is actually not a Swedish company, but from our friendly neighbors in Finland. Welcome, Paul.
Thank you.
Take it away.
Okay. So, BBS is a Finnish biotech company. We make a product that's quite unique. It's in the bone graft substitute market space. It's based on pioneering research on bone healing by the company's founder, Professor Pekka Jalavara. The company is headquartered in Oulu, Finland. We have a manufacturing site in Reisjärvi. We have 12 employees. We're planning an early commercial launch of our product, and we're in the planning phase now. So, Professor Yalavara is a professor emeritus of orthopedic and trauma surgery at Oulu University, founder of the company. While at Oulu University, he established the bone transplantation group, which had an objective of finding an alternative to autograft. Autograft is a surgical procedure where the surgeon performs a second surgery on the patient, usually at the hip bone. The problem with this is that it increases risks and complications, and the patients often have up to 40% of these patients have long-lasting pain, significant pain following the second surgery. So the bone transplantation group was set up to examine other animal sources for using bone extracts, and found that the reindeer bone extract had a significantly higher degree of bone regenerating potential than all other animal sources. So that's the start of BBS, and we're ready to take to the market our ArtBone medical product. So, why reindeer? Reindeer have a significant capability of regenerating new bone, about 30% of their body weight every year, both males and females. And there's two required components for effective bone healing. One is osteoinduction. This is just simply to start bone growth, and this requires proteins. This is where the reindeer comes into play. Osteoconduction is the other component that's needed, and this provides a surface for the new bone to grow. The osteoconduction component is provided usually by a mineral like calcium that provides a scaffold and a surface for the new bone to grow on. The reindeer bone protein contains biologically active proteins and growth factors to provide the osteoinductive component. We also include a synthetic beta tricalcium phosphate, which is a mineral component, to provide the osteoconductive signal. The other function of the reindeer bone proteins is to accelerate the resorption of these TCP granules to advance faster and better bone healing. This is a demonstration of how the ArtBone product works in a large animal model. This is a sheep, and I won't go through the histology on the left, but just simply summarize the results. Within 3 weeks, more new bone was grown. Within 8 weeks, a good quality bone, increasing resorption of the TCP granules, and by 16 weeks, there was complete bone formation in this model. This was the last animal model that was needed before the company could then proceed to human clinical studies. Here's an example of one of the patients in this study. 34 patients were enrolled in an ankle fusion study. This is actually the first in human implantation of reindeer bones protein extract, and the ankle on the left is before surgery, and you can see with the dotted line on the right-hand side here, a complete fusion of this ankle space. This was the last and only needed clinical study to present to the notified body in the path towards CE marking. So our first product is ArtBone, including the reindeer bone protein extract and the TCP granules with glycerol and other excipients to form a flowable paste in a custom designed syringe. This is ease of use, one step, no mixing required for the surgeon, and this particular formulation is ideally well suited for foot and ankle, wrist and hand, small joint spaces, but it can also be used in other areas of the body as well. We'll expand our product portfolio by coating TCP granules with the reindeer protein extract, as shown on the bottom, ArtBone granules and larger ArtBone blocks. What this will do is expand the orthopedic application of this product for wider uses and to gain a wider market. So this is the market position of the ArtBone family products in the bone graft substitute market. The autograft still comprises about a third of the clinical procedures done, still considered the gold standard. The bottom segment there, the demineralized bone matrix and allografts, are all derived from human donor bone. Significant problem with this segment is that the FDA requires that each lot of product be specific to each human donor. So these products vary widely in quality and consistency. The synthetics group are the mineral components like the TCP that we use. And the other section there is the recombinant growth factors. This component is a drug type product and is very potent, but also comes with serious complications and side effects. The chart on the right here shows that the BBS Heartbone product is in the sweet spot with respect to autograft in terms of its osteoinductive and osteoconductive capability. So the current bone grafts substitutes market in the 10 major markets is worth more than $2.9 billion, is expected to grow at a rate of a steady rate of 4.3%. We've mapped the surgical procedures done in the United States, about 1 million, and in the EU5, 440,000, and the sales revenue attached to these procedures in both the US and EU. The worldwide market opportunity for the Heartbone product family, based on selling a dose of around $1,100 per dose, retains a profit margin between 80 or 90%, and gives us an opportunity to look at reaching a potential large market. On the right-hand side is the 10 major market players in the orthobiologics product market. These companies account for about 64% of the market revenue in the orthobiologics product space, and are also looking at small companies for innovation. So Heartbone is expected to compete effectively across all market segments for the following reasons. Safer than the recombinant growth factors. Also are safer with respect to the machine banked and DBM allograft products. Makes up for the significant weaknesses with these products, since they're derived from human donors and are weakly performing. The synthetics are is a group that our TCP comes from, and these products are also have safety issues. Go on. The patent portfolio is broad and strong, and we have granted patents across the United States, Europe, and Asia. We have a manufacturing facility in Reykjavik where BBS has received a certificate of GMP compliance from the Finnish medicine agencies for manufacturing of this biological protein extract. Where the company is prepared to get ISO 13485 certification for the manufacturing of Heartbone medical device. Our current production capacity is 25,000 doses of Heartbone per year, and our maximum capacity can reach 500,000 doses based on the number of reindeer that are slaughtered in Finland every year. Heartbones are ready-to-use paste as a final product assembly, and we control the assembly process in-house. So I'll summarize just briefly the different stages that the product has gone through. This is typical for a biopharmaceutical development pathway where significant safety and toxicology studies were done on the reindeer protein extract. Preclinical studies were done in large animals, and human clinical study done in the ankle fusion study. The product certification is ongoing. So our development timeline for Heartbone is shown here. We're looking at a CE marking to be achieved by the middle of next year. We have a pre-submission process in place with the US FDA. Once the CE marking is obtained, we'll be able to begin product launch in Europe, and then with our Artbone product extension in green, we expect product development to follow pretty much the same pathway. Our sales strategy is to start by introducing the Artbone in syringe product into the Nordic markets. We'll use sales specialists, a small direct sales team. They'll have the added advantage of being able to introduce this product to surgeons based on the clinical validation already done in the clinical study. From there, we'll also develop distributor partnerships with knowledgeable distributors in selected European countries. We have a strong management team and a board team and scientific advisors with a broad base of technical and business background. So the investment in BBS is based on a strong value proposition associated with the fact that the Artbone reindeer protein extract provides a significant advantages and opportunities over existing market segments, and the market for the bone graft substitute markets is large and steadily growing. We will look to get the CE marking from Europe, from the notified body within a year, and then advance to marketing the product in the United States. We have a fully built manufacturing plant that can control the product from supply to final assembly, and we're looking forward to welcoming participation. Thank you.
Thank you very much, Paul, for a very interesting presentation. So, reindeer, huh?
Yeah, I should reframe the entire presentation. I'm sorry for that, but reindeer the reindeer is actually a unique resource because the animals generate about 30% of their body weight in antler every year and drop it. One of the key discoveries that Professor Yalavara made was the fact that this has kind of nature's bone-making machine. And so the Artbone products basically have captured the reindeer proteins, which are essential for providing the osteoinductive signal. This is a component that is exclusive to BBS, and the bioprocess technology behind it took 10 years to develop. It's actually a very sophisticated biopharmaceutical process. The reindeer proteins have been certified by the Finnish Medicines Agency as a medicinal substance. So this has a development history and a quality profile behind the product that is unmatched. Most other bone graft substitute products that go to the market don't have clinical validation, and here BBS has a claim for basically having the first in-man studies done, demonstrating the safety and the performance of this reindeer protein.
So that would be the unique selling point.
That's a very strong selling point, and the other point is the fact that the device, this is classified as a medical device class 3 type of a product. It's highly innovative and would be expected to be received with enthusiasm, especially by younger surgeons that are looking for innovative technologies.
And then looking to tap into this $2.9 billion market that it totals, what would be the main challenges to ensure a good market uptake for BBS?
Well, a big challenge there, of course, is introduction of highly innovative products, which could have been better presented to the audience today. I apologize for that. But the challenge, though, is also going to be strongly met and supported by the fact that, unlike most products that come into this market space, they go directly into the market. In BBS's case, it has a strong clinical safety and performance validation already behind the product. So that'll make it easier for the product specialists to get the attention of orthopedic surgeons. Also, Professor Yalovara's network, the clinical directors of the 3 sites that actually evaluated the product in humans, will also be key opinion leaders and help support the product launch and entry.
And the market strategy is to first look at the Nordic countries and the European market?
Right, correct. As a small company with only 12 people, it's quite remarkable development history, and I didn't do a very good service to representing that today. But for such a small team to take a highly sophisticated medical device class 3 product that's actually going to serve as a best-in-class product once the CE marking is obtained is an extraordinary achievement. And so that the value proposition there is that it will have an opportunity to replace the existing market segments that are now being used as bone graft substitutes. And I'll probably have enough time to make up for the lack of sufficient communication on the slide presentation, but I can make this point quite compellingly now, and that is that no other bone graft substitute product in the market has what the ArtBone product has, because it combines both of these principal components in one integrated medical device. It both provides the opportunity for new bone growth to grow with the TCP chemical component, and the Ranger bone extracts, unlike any of the other products, are biologically active proteins, including growth factors. And so this is the component that provides the osteoinductive signal to start new bone growth. So the value for this particular product is it can actually generate new bone faster, better than existing products. The DBM, demineralized bone matrix products, are just basically kind of a little slightly more processed version of an allograft. An allograft and DBMs, though, both come from human donor sources. The problem with the human donor sources is that, and the FDA requires that each of these products be lot specific to these donors. The problem there is that they vary widely in quality and consistency because of health and age issues. One of the things that the existing products can't do, and that only BBS can do, is basically pool large amounts of reindeer bone and then guarantee to drug level standards the consistency and quality and performance of each large batch of product. So this will actually serve to be, in fact, an early replacement candidate for already billion-dollar market segments of the bone graft substitute market. So we expect we can get by with a small focused sales team because they'll come armed with a value proposition that can't be matched by any other products in the market, especially the DBM and the allograft products. These should fall pretty quickly because they've already been shown to be widely inconsistent in the range of their osteoinductive capabilities and the kinds of bone growth factors that are in these.
So entering a fairly crowded market competitive-wise with an aggressive value proposition like that should stir up some commotion in the market.
Well, in the medical technology space and the orthopedic industry space is very well known to protect what they have, but the big players also look to the small players for innovation. And so the BBS ArtBone product fits very, very nicely into a position where, depending on how well it performs once it gets into the clinic, the opportunities are basically for BBS to decide where to go from there in terms of either expanding and becoming an independent, full-fledged sales and marketing entity, or the big players already are looking at BBS because they have an eye on the degree of innovation that's behind this product.
And in terms of strategies, would you say that the company is leaning towards one or the other, as looking at the prospect of being acquired, for example?
Well, I think it depends on the timing and the value of the offer, but usually what happens in these cases is more likely a strategic alliance or a partnership. Large companies like to develop certain types of distribution arrangements with a company and groom a smaller company and that way the values increase to on both sides, and possible exit by acquisition is common that way.
I should ask if anyone has a question in the audience.
Yes.
Yes.
You're introducing reindeer protein into the body of human body. Do you see any immunological reactions, any antibodies produced?
Yeah, this was a very important question, and in fact, this was something that was addressed early on in the history of the product development because it's developed as basically as a medical substance. It went through a full set of safety and toxicology studies. It's been in a variety of small animals from mice, rats, rabbits, dogs. Full safety and biomaterials, biocompatibility studies have been done. So with respect to humans, and by the way, along the way there were no indications of an antibody reaction, which is usually shown by increased inflammation. And so the preclinical studies that had been done were enough to qualify through institutional review boards the advancement of this for first-in-human studies. And so those studies were conducted for safety and performance outcomes. And although we're actually in the final stage now of actually summarizing and finalizing a clinical report. And the good news is that there's no evidence in terms of inflammatory markers. And so there's a good reason to believe that there's not going to be any immunological issues associated with this product.
Because if you get antibodies, you might not have any big problems with it, but maybe the antibodies present that will be generated will prohibit activity for any next time treatment.
Absolutely, but then in this case, these types of products as bone graft substitute implants are usually just done once.
Yeah.
And so there's very little risk for a potential immune challenge with subsequent doses. If there was, this was a different type of a drug product, then that would be a consideration. The other thing is fortunately, the immunological reactions are actually stimulated more by bone surface. And so whether in the processing of the reindeer protein extract, there's a long process that's gone through to basically demineralize the bone and then concentrate the bone proteins and the growth factors that are needed for regenerating new bone.
I'm sure there are more questions floating around. We're going to have to take them after. As of now, I thank you very much for your presentation.
Thank you.
Thank you. So, from healing bone to preventing thrombosis or blood clots. Next company to present is Cereno Scientific, and I'm happy to introduce to you Sten Sörenssen, CEO of the company. Welcome, Sten.
Thank you, Jonas. All right, so I'm happy to be here and to present Cereno Scientific's vision, our significant progress the last year, and also our plans for the future. I hope you have some good questions for me when we're done. So our vision is to develop a first-in-class drug to prevent thrombosis. Thrombosis is the #1 killer on the planet. And there is a huge need for better efficacy and also to avoid the risk with the current therapies. Looking at who gets thrombosis and what the impact it has, so you can see that here is about half of the deaths are caused by cardiovascular diseases, and most of that is thrombosis related. So stroke in the brain, infarct in the heart, thrombosis in the veins. And here you see, so it affects the vital organs mostly. And you can see the background for thrombosis is the welfare diseases, so to speak, smoking, diabetes, overweight, inflammation, and so on. So it's very common, and it's the most common cause of death. Here you see, because it's so common, you can see it's a huge market for therapies. And it's a bit old data here, 2017, but around $20 billion per year and it has a growth rate to double within 7 years. So by 24, it will be $40 billion. And that's the market we're going into, and we're going to hit the market about 24, 25 approximately with our drug. Now, what's used today? You probably know this, and it's called blood thinning drugs. Anticoagulants or antiplatelet drugs, two different kind of blood thinning drugs, so to speak. And the reason they're called blood thinning drugs is that they affect the coagulation system in the body. So it actually makes it harder for the body to coagulate if you have a bleed. It prevents thrombosis pretty well, but it could be better. But the major problem here is that you see the man here is red, all the vessels are red, and it's to illustrate that if you take these drugs and you take them every day, most people for life after an infarct or after you had a stroke. The problem is that you are at risk of a bleed, a major bleed or any kind of bleed 24/7. So every minute or hour you are on these drugs, you have a risk to experience a bleed. And it can be a fatal bleed, it can be a major bleed, or it can be any kind of bleed. For instance, nosebleed that doesn't stop. You have to go to the hospital. So all drugs act as blood thinning drugs, and it's a systemic drug effect. Now, it increased the risk of bleeding, as I mentioned, and the problem with that is then that the pharmaceutical industry has developed these drugs in lower dosages than would have been the case if they didn't cause bleed. So if you increase the dose, it's high risk for bleedings. So there's both an efficacy problem on the market and a risk problem, adverse reaction problem. So there is a need for new preventive strategies on the market. Okay. How common is this? Well, major bleeds are, and this is in clinical material, up to 2.5–3% get major bleeds, dangerous bleeds in stomach, brain, and up to a quarter in clinical materials get any kind of bleed. So every fourth patient. And this is in clinical material where the most risk-prone individuals are not included. So, big problem. Now, this is to illustrate the body, and the body has two different ways of dealing with bleedings in the body. So you see to the left, there's a coagulation system, so the body can coagulate. So you stop a bleeding on the skin, for instance, you can see it. It becomes a little coagulation and a scar. And the body also has another system called the fibrinolytic system. So we are equipped with a system to dissolve blood clots. All right, thank you. So we are equipped with that system like a high-rise that has sprinklers. You know, if there's a fire in a room, the sprinkler in that room goes on and water comes out. That's how our defense mechanism works in the body, and I'll illustrate that. And if you are a risk patient, that doesn't work. Now, as I mentioned, all the drugs on the market are blood thinning. They act on weakening the coagulation system to prevent thrombosis. We are acting here on the fibrinolytic system. So we are restoring that system to become better, a better defense than risk individuals have. That system is too weak. And let me show you how it works. So this is a vessel. It's a plaque, atherosclerotic plaque that most of us have in the room here, I think. And when you get older, they can burst, and then you get a bleeding inside the vessel. And when that heals, you get a blood clot forming, and you don't want it to occlude the blood flow. When that bursts, there is a signal going up to the cell wall inside, and it releases this fibrinolytic water, so to speak. It's a protein, and it dissolves the blood clot so it doesn't occlude, grow and become too big and occlude the blood flow. So it's a very important safety mechanism. There is another protein in the blood floating around called pi, And the main activity of pi is to block the action of TPA that's coming out. It's a balance system. It's all good if it works, but in risk individuals, the TPA, the sprinkler, there's no water in it, so to speak. There's too little of the protein, and there's too much of the floating pi. So you get too little coming out, and what's coming out is blocked by pi. And then the blood clot can grow, and you get an occlusion, and you have an infarct. So that's very bad. So these are two risk factors for myocardial infarction and stroke. Just very quickly look at pi here, the floating protein. That's very high in the morning hours with risk patients, and that's exactly when it's most experienced to have stroke or infarcts in the morning hours. And you can see that to the right on the black staples there, angina pectoris is a coronary event, and there's acute myocardial infarction. You can see the hours correspond with the early morning hours, somewhere between 5 and 9 in the morning. So that's when you want your defense mechanism to be intact. And look here at this slide. This is a heart and a coronary vessel, and you see to the left, you see it's working. So you get a small bleed, TPA is showered out, the sprinkler, dissolving the clot so it doesn't become too big, and pi blocks the rest of TPA and takes it away, and it's all controlled. If you look to the right here, you see a risk individual. There is very little TPA in the cell wall. There is a bleeding starting, and the blood clot is growing, and pi is in high concentrations. Let's say this is in the morning hour, and you get an infarct. The defense mechanism is too weak. And as you can see from the heart, downstream there, it's all white. You see the heart. That's necrosis. That's dead heart cells. And that's what happened in a myocardial infarction. So you either die or you get an incapacitated heart. You can develop heart failure and so on. So you want your defense mechanism to be intact. What we are developing is a product, CS1 we call it. It's our lead project. And that will make possible a restoration of the defense mechanism. And I'll show you how. So we will increase the synthesis, the storage of TPA and the release of TPA, and will also reduce pi in the morning hours. And I'll show you the documentation we have. And that way, if you take this in advance every day, you will have a defense mechanism that's stronger. So we have a dual mechanism of action. Our product can increase the synthesis, storage, and the release of TPA from the cell walls, and we at the same time reduce the risk factor pi from the bloodstream in its concentration. And by that we have a stronger defense mechanism. And the beauty with this, if you look to the right in the picture with the arrow, it's activated only when and where the body needs it. The body itself activates this. So we are not causing a situation where you have an individual that are at risk of bleeding 24/7. So it's a very different approach compared to what's out there today. Yeah, so the advantage with this first-in-class drug, CS1, is that we prepare the body to have a good defense mechanism against an event. And we restore the fibrinolytic capacity, and it's activated only when and where needed. So you see this man here to the left, or woman, is not all red. It doesn't have a risk of bleeds 24/7. So our objective is now to develop this drug and to use it as a preventive therapy. And we think it's going to be very valuable as an alternative to the existing drugs out there. It's going to be safer and it's going to be effective. And the major indications are deep venous thrombosis, myocardial infarction, and stroke. And prevention of stroke is the major indication here. Now let me just quickly go through some of the data that we have to support our vision and our development program. So we have in vitro studies, so cultured human endothelial cells. We have animal models in mouse and pig. I'll show you a mouse model today. and the data. We have human physiological data, and I'll show you that. And there's also very interesting epidemiological studies indicating that this really works in man already. And then we have the data on our phase 1 study that we concluded last summer with very positive data. So first, the in vivo data on cultured cells. And here you have two pictures. One is without treatment and the other one is with the active substance that is in our drug, CS1, valproic acid. And you can see the green stains here are actually TPA. So TPA in the cell wall. And remember that I mentioned that that's one of the effects, is that if you take this in advance, there will be much more production of the protein that you need to have in the cell wall. And the green stain is staining that protein. You can compare the pictures and you can see there's much more. This is our proof of concept data in live animals treated for 3 days, 72 hours. And you can see here on the x-axis is time and on the y-axis is the fibrinogen content. And these mice were hurt in a vessel, a vein, and they were pre-treated and hurt, and then there is a bleed and there's a thrombosis formation. And the treated group is the red line and the control group is the black line. And you can see already after 2 minutes, in the very beginning there is a significant difference between the curves. So there's much less thrombosis buildup in the mouse that was pre-treated. So it has a defense mechanism already against infarct, if you will. And the photograph shows the size of the thrombosis in the two, treated and pre-treated. And you can see it's much less in the lower picture. Now let's move on to experimental studies in man, and here are 2 pictures, and they show that the substance in our drug has an impact on the 2 biomarkers for infarction or thrombosis. The left picture is a forearm model treated over 4 weeks, and it was documented that we release TPA better if you are pre-treated. It's the top line. So there's more TPA released in the humans. And they were people with coronary disease or heart disease. On the picture to the right, there is an effect, a study on over 2 weeks to show the impact on the other important protein here, the risk factor for thrombosis, pi, the one that was floating around in the body. And here we saw up to 50% reduction of pi in blood. So we have documented in man impact on these 2 markers. Now let's move on to epidemiological studies. There are actually 3 different publications. There are 4 here, but there are 3 different studies. One Danish in 2011, another Irish that confirmed the first one in 2014, and one last year, 2018. And it's research groups that have looked backwards into patients that have epilepsy. And the reason they're looking at that is that the substance that we have found is already used for epilepsy, has been used for 45 years as a drug for epilepsy. So they have gone backwards and looking at, does it matter what kind of drug you are on for cardiovascular incidents, myocardial infarction and so on. And all of these studies has confirmed the indication that it matters. So if you are on this substance, VPA, which is in our drug, you have less infarcts and you have less stroke. So already in long-term studies, and I'll show you one because we don't have time for all. This is the Danish study published in 2012, 150,000 patients were follow-up with epilepsy. There were a variety of drugs. One of them were VPA, used for epilepsy, 10 years follow-up. And the difference in myocardial infarction in the data material was almost 40% less myocardial infarction, only in the group that had VPA. Similar kind of data is for stroke in the latest study from 2018. So it's very encouraging that this data already exists in man. Now, let's move on. So we have developed this VPA into a very specific formulation. It's an innovative concept where we have a delayed immediate release formulation of VPA. So if you take the tablet, it prolongs the release of the drug, and then it releases it. And the reason we've done that is that we want to affect pi in the morning hours, as we spoke about before, as I mentioned. At the same time, we want to have an effect on the TPA production. And the data was, we published the data, presented the data in June last year. And no problem with safety and tolerance, and we didn't expect it because the drug is already used in man, the substance. We were looking for these pharmacokinetics with a specific profile in the blood so we could affect pi the most. And we actually found that pharmacokinetics well. We used 2 different dosages. And what's not normally done in phase 1 trials is that you look for anything else than safety and tolerance. But we looked for an impact on pi, and we documented a significant reduction of pi in the morning hours with CS1. In addition, we also documented bleedings, and there were no increase in bleeding in this study. So that was very encouraging, and we had already 8 months previous to that started to do a procurement to find a CRO to do our phase 2 program. And we changed our strategy from doing a small phase 2 program to doing a really big, I would say, phase 2 program for a company our size. And the reason is that we want to do it the same way that the big pharma has done proof of concept in man. The products that are now on the market selling for most part of the $20 billion have done this kind of study that we will do. So we signed a contract with OCT. So we will do a study in circus 30 centers, multi-center in Russia and Bulgaria. And we'll do it in orthoplastic surgery, actually total knee replacement. And the reason is that when you go through that kind of surgery, the risk of thrombosis, blood clot, is increased. Other good news during the fall here was that we got a major good news. We got patent approved in the US, which makes up about 50% of the market. So that was very positive. Other news that we got was, or what has happened recently, is that we have terminated our convertible bond financing that we signed a year ago, and we are pursuing other financing. I'll come to that at the end. We also got a patent granted in Australia this spring, another important market. And then we have established a very strong scientific advisory board. I'll mention it at the end here. We're working very actively at the moment. A couple of other events. We acquired a new substance from a group at AstraZeneca. Used to be at AstraZeneca, but they're at the BioHub, where we are also located in Gothenburg, AstraZeneca's facilities. Emeriti Bio. They are the group that were behind Losec and Losec's follow-up, Nexium, Brilic, Plendil. Those are the chemists, and they have formed this company. And we have now acquired one substance from them that fits very well with our concept. This was done in March, and we filed quite rapidly a patent here in May on that compound. Yeah, so I will jump this. So we are developing a new preventive therapy for thrombosis. Reasonably short time to market because it's been in man already, the substance. We have a double effect, TPA synthesis, reduction of pi in the blood. We have documented this in vitro, in vivo, experimental human studies, epidemiological studies, 3 of them. We have a positive phase 1, and now we're pursuing phase 2. We got patent in US and Australia, and we have expanded our pipeline. Briefly mention something about scientific advisory board. These are heavy cardiologists and researchers. I'm going to meet with them in Athens this week, in connection with the European Society of Cardiology's annual heart failure meeting. And so Dr. Bertram Pitt is chairman. I've worked with him in the past in major studies. And Dr. Gordon Williams is from Harvard Medical School, so is Deepak Bhatt. Professor Fayez Sanad is from France, runs the cardiovascular clinical trial program that FDA used to run in Washington every year. And Gunnar Olsson has been one of the heads of AstraZeneca's research for many years, both cardiovascular and gastro. Now, upcoming milestones. So we are imminently going to submit our phase 2 study application to authorities for approval. We thought we were going to do it in Q3, but we've moved it backwards to Q2 because it seems that we are ready. So look out for that news. We are pursuing a share issue very shortly. I'll mention that at the end. And so we expect the authorities to approve our phase 2 during the fall. We hope so. And then we are scaling up the production of our clinical material, including our drug, for that study. And that's a hefty job to do. And we're actually working at it at the moment. And we expect to start the study during the spring and to complete it by the end of 2021. So the study takes about a year to do, and then there's another 6 months before and after. So end of 21. That's it.
Thank you very much, Sten. Very interesting to see all the things that you have accomplished the last year. Preparing for a phase 2 study. You said that this study is designed or is going to be made the way a big pharma would do it. What's the reasoning behind that? Because I imagine that makes it a little bit more expensive than it could have.
Absolutely. So yeah, it's if you have big visions, you have to do the job. So and we have, we're very skilled people in our group and external advisors, and we adjusted our strategy to do it the way Big Pharma does it, because we don't want the data to be questioned, and we want to make the best effort possible to document proof of concept in man to prevent thrombosis. So that's why, and it is a bigger price tag. So yeah.
But you gain some in the end, hopefully.
Absolutely, we hope so. Yeah.
So in the end, you mentioned the new scientific advisory board. Like you said, there are quite distinguished names on the board. What has attracted these people to Cereno, you think?
Well, firstly, I have started a couple of initiatives in the past that's been going very well. Survival trial in heart failure and a new concept with aldosterone in the 90s. And Dr. Pitt, who's chairman of the scientific advisory board, was actually the chairman of that survival trial, which was published in 99. So that went very well. And Fayez Sanad was also in that study, and in the next study, which was started together with colleagues when I joined AstraZeneca, I initiated with them a survival trial with Astra's beta blocker, Seloken or Toprol XL. And these two studies were published the same year, and they were very positive. So now when they got to know what we were doing here, these are very busy people, and they go for science. And actually, they wanted to work with Cereno.
Yeah.
So yeah. So when they got to know what we were doing, and you will later see the results of our work together. We formed this scientific advisory board this spring, and we're meeting now. But we've already started working, and I will be happy to share more of that in the coming months with you, what we're doing.
Yeah, great. We are running out of time, so I'm going to let one question from the audience.
Is your substance CS1 identical with valproic acid, or is it a modified version that has implications for the studies you have to do and the size of the studies.
Yeah, exactly. So the substance that we have in our drug, CS1, the chemical substance is identical. So what we have done with that is we have reformulated. It's quite complicated formulation that we have developed together with Galenica since late 2006. Beginning 2016. So it's the same substance. So it's the same substance that you see in the epidemiological studies and so on, but different dosages and very different formulation. So the plasma profile of it and the concentration in the body is very different. And the reason is that if you give this for epilepsy, you need to have certain concentration to prevent the grand mal or whatever is happening in epilepsy. But in this indication, what we are after is stimulating it's an epigenetic stimulation of TPA synthesis that takes some time to get going, and then there's a constant reduction of pi in the blood that we need to get done. And for that we need different dosages and different profile, pharmacokinetic profile. And that's what we have developed. So we have filed with 3 patent families. So we have filed usage patent first, and then a patent in relation to pi and thrombosis, and then a patent in relation to the formulation. And we are in national phase with the second patent family in 21 countries at the moment.
Because it has implications for the safety. Now, the phase 2, how many patients are you expecting in that? And the following question, how many would you think are needed for a phase 3?
Well, yeah, so I can't really tell you how many we have in this study yet because I haven't published it yet. We haven't released that information. And it depends also in relation to our discussions with authorities on a clinical protocol that we are submitting. But it's 30 centers, and it will be several hundreds patients that we are pursuing. And you're very right. I think the safety for new chemical entities, you never know what's going to happen. So if you do small studies in the beginning, you might get hit by a side effect in phase 3 in a major study, or even in phase 4, like COX-2 inhibitors, for instance. They were pulled from the market. In our case, this has been in man for 45 years. So it's quite well known. It's been in much higher dosages than we will use in our drug indication. So we expect less documentation needs and absolutely much less risk. And so again, several hundred patients in these studies. And in the phase 3 studies, I have to return to you with that. We have been looking at it. So it's not small studies. All right.
So we will be looking forward to the release of more information once you have sent in the application for the phase 2 study to regulatory authorities. Yeah.
I have one more thing I'd like to say, but
yeah, we can do that in a little while.
Yeah.
First, I'm going to thank everyone for watching in the US, and thank you everyone for coming. Nu byter jag till svenska. Sten har en slide till som han gärna vill visa.
Det är ingen surprise kanske, men vi terminerade ju vår financing agreement för fonden som hjälpte oss fram till där vi är idag. Och nu har vi precis releasat vi vårt prospekt igår för en share issue, en emission som vi 23 maj till den 7 juni så är det teckningstid för den, och den är ju till för så att säga de som har aktier redan. Men 2,90 är teckningskursen och vi planerar att ta in 55,6 miljoner. Den är garanterad till 80% och management har puttat in 6,5%, 3,6 miljoner. Där är en overallotment också, om det skulle vara många som tecknar sig så tar vi in 5 miljoner till. Och detta är för att ta oss fram till och med start av fas 2-studien. Sen behöver vi göra en finansiering till för att genomföra hela fas 2, för den kostar så pass. Den här ekonomin är också till för att driva patent och andra saker, men också för utvecklingen av CS014 och nästa substans som är i preklinisk fas, den jag berättade om. Så har ni aktier så teckna gärna. Ni var med på resan.
3 snabba argument för varför man ska vara med på den här resan fortsatt.
Ja, alltså ett argument är ju, huvudargumentet tycker jag är om man verkligen tror på teknologin. Jag är en teknologiperson, jag går på teknologi. Den andra kan ju vara mer finansiell, alltså att ett bolag som är i vår fas är normalt sett, man ska inte prata för mycket om värdering, men normalt sett hyfsat värderat på börsen. Vi är värderat till 60 miljoner kronor ungefär. Och vårt börsvärde sjönk med 80% under den här finansieringen med fonden som var convertible bond som tryckte nya aktier och sådär var 6:e vecka och släppte ut på marknaden. Och den har vi då terminerat så att jag tror att vi har ett nerpressat börsvärde idag på 60 miljoner. Det skulle varit kanske 200-300 i nuläget. Och den tredje är ju att om man ska man ska ju helst in innan en fas 2 görs. För när den blir färdig, om den är positiv, så lär det få ringa på vattnet så att säga.
Nyckelord, om den är positiv.
Ja, absolut. Så är det ju med all forskning.
Kan du säga någonting bara om den här CSO14? Vad är det för någonting?
Nej, inte mer än att den är innovativ. Faktiskt. Så att det är inte så mycket än. Och det finns flera skäl till det. Det är patentrelaterade skäl och det är också konkurrensskäl. Så att vi avvaktar lite med det tyvärr, får jag säga så, denna gången. Men vi hoppas komma tillbaks med lite information kring den om möjligt här under hösten. Men det är en spännande substans, absolut.
Det ser vi fram emot att höra mer om. Tack så mycket Sten. Andra frågor får vi ta efteråt. Tack så mycket.